Right, next speaker is Mancuso from Pasanic Lab, and he's going to be telling us about TWAS, also discussing transcriptome-wide association studies.
Yeah, thank you for the introduction. So today I'm going to talk about a tool that our lab and Alex Price's lab has been working on called TWAS. But first, let's just do a brief background over GWAS, and that is typically GWAS maps risk for disease, and the way that works is you genotype tens of thousands of individuals for large-scale studies, and you have a disease status, 0, 1, indicating whether or not they have the disease. And you go ahead and scan the genome, or the genotype rather, and look for associations between genetic variation and disease status. So this has been particularly useful for mapping regions of risk for various disease. However, one of the problems with it is that it doesn't really give us any sort of biological insight as what is driving the risk at a mechanistic level. And so one of the things that we could do is possibly look at various intermediate phenotypes, whereas with gene expression being a good example. So here we have gene expression measurements for a bunch of genes for a subset of individuals, and there are two things that we can do. We can then go ahead and perform an EQTL study where we look and see is what genetic variation drives changes in the abundance of various transcripts. And the other thing that we could do is go ahead and associate the gene expression itself directly with disease status. The problem with that second approach is that it's going to be very noisy. There are a lot of environmental factors that may influence the abundances of gene expression, and we can't exactly pinpoint what truly is a genetic effect. The bigger issue, though, is that gene expression data is very expensive to collect, right? So it's typically limited to hundreds, if not maybe a few thousand individuals, compared to GWAS, where we have, again, upwards of tens of thousands of individuals. So it would be nice if somehow we could either get a bunch of money to fill in that gap by collecting those samples, but there's an alternative approach in that we could simply just predict the genetic component of expression or impute the genetic component expression for these individuals, and then go ahead and proceed with our associations between expression and disease status for individuals. This imputation expression can be performed under various ways, but what's kind of neat about it is that under this model, where we go ahead and impute gene expression into large panels and then test for association with disease, you can think of a sort of a causal chain, right, where we start with genetic variation data that influences gene expression, which then influences trait. Now, this is able to distinguish from non-cis-regulated correlations, right? So if it's not hard to imagine someone who has disease status, and that in turn affects their expression levels due to various medicines they may be taking, other influences. Again, these two kind of graphical models don't paint the entire picture, but it does give us some sort of high-level insight on what we're able to test. So the tool that Alexander Gusev and Bogdan's lab developed was called TWAS, transcriptome-wide association. And one of the things that we can use for it is, one, new locus discovery, right? We can aggregate effects into a single expression instrument, which results in fewer tests, right? This time we're testing at the gene level for association rather than just individual SNP level. And also fine mapping, right? Genes are much more interpretable units as opposed to the individual SNP data. Again, the one maybe, depending on how you look at it, downside is that this does ignore effects that are entirely non-genic, right? So these are only looking at the effect of the genetically driven component of this expression, more specifically the local, right? Like a cis sort of effect for a given gene. So this isn't the first approach to do that. There have been a couple other papers. So Gamazon et al. Has a method called PredictScan that employs this sort of reasoning when you have individual level data. Alexander Gusev's paper, that's what we're talking about today, TWAS, is a little bit more flexible approach. And then recently there was another paper that looked at summary statistics data from both GWAS and EQTL studies. So today I'm just going to focus on our method, TWAS. So let's just start briefly with the model. We can think of gene expression as a linear combination of SNPs with some environmental component. So given genotype data for a panel which we have gene expression, we can then fit this model. You can either use linear regression, you can use various regularized approaches. We typically tend to use BLUP that takes heritability estimates into account to kind of regularize these estimates. And once we obtain those, we can then take the external reference panel for which we lack measurements, right, and take our weights and then go ahead and predict or impute the cis component of gene expression. So once we have that component, then we can go ahead and test for association with disease status. Now, does this work? Well, so in Sasha's paper, he went ahead and looked at fitting this one particular model to gene expression that takes the entire region into account. And it shows that there's considerable gain in using the entire cis window as opposed to just the top EQTL. So what that indicates is that there's pervasive allelic heterogeneity going on, right? That there are typically multiple SNPs that influence expression at these genes. We do see a lot of the time, right? Maybe about half the time that they're both perform equally as well, but there's a long tail of performance to be gained by considering the region as a whole. And so here we have 3 different studies that weights were learned from. We have METSIM, where we had gene expression measure in adipose tissue from 570 individuals, the Young Finn study, where we had 1,400 blood samples, and then the NTR study, with about 1,200 blood samples. And they all showed pretty similar performance patterns here. So the TWAS, so if we went ahead and looked at how well we can predict the component of expression under either a single EQTL or using this Bayesian sparse linear mixed model, in this case, we see that TWAS performs better 1.3 times on average. And of course, we get better performance as sample size increases, which isn't surprising. Now, one of the cool things that we notice is not only can you go ahead and perform this imputation when you have individual level data, but you can go ahead and do it directly with only summary level statistics, right? So it makes sense to just impute the z-score itself that would measure the effect of the genetic component of expression on a disease status. So if we model the GWAS summary statistics that we have from a large-scale genome-wide association study like the GIANT study for height, for example, right, we're interested in imputing this value here, right? The z-score that indicates kind of the strength of the covariance relating gene expression to the initial trait. So you can approximately model that under a normal assumption, where all we need is LD estimated from an external panel, and then the covariance estimated between these SNPs and the gene expression. So all that says is that this z-score here is really just a linear combination of the GWAS z-scores under certain assumptions while we take LD into account, right? And not only that, but we can, under certain circumstances, this model is equivalent to just estimating the genetic covariance between the cis regulated expression and the trait of interest or disease of interest. So when we look at the performance from the individual-based method along with the summary-based method, we see a correlation of nearly 1. So there's not much power loss by using the summary-based approach. So this is really, really cool. And now that we can be, what was the phrase, like data parasites, right? We can go and take these massive summary statistics and perform this imputation using weights that are provided, that we provide on our website, to see what the genes of interest are. So we went ahead and did this with 30 complex traits. So this is work in progress that I've been working on with Sasha Gusev and my PI, Bogdan Pasanic, along with Huembo. So we looked at summary data that comprised over 2.3 million phenotype measurements across 30 traits. We tested over 9,000 genes over 40 tissues. So the tissues were mostly from GTAC. We also looked at the ones I mentioned earlier, along with Common Mind. And so here we have the traits listed along this axis, and the number of significant genes that either overlap a genome-wide significant SNP or do not overlap a genome-wide significant SNP. So the ones that lack the overlap we consider to be novel in this sense, and that there are probably multiple allelic variants driving risk there, and individually they don't achieve significance. Right, so we have a massive gain with height, which isn't surprising considering how polygenic it is. But there's certainly a lot of interesting things to be discovered using this method. Another thing that we kind of looked at was how pervasive are pleiotropic genes, right? So again, we just looked and counted and said, all right, for a given gene, how many traits does this turn up to be significantly associated with? And so again, there's a nice tail here where most genes, and this turned to be were associated only with a single trait. However, quite many were associated with multiple. And so here, this is a confusing plot where we have genes ordered by chromosome and the traits by track, indicating that they were associated there. So again, this is work done with Sasha Gusev at Harvard School of Public Health, along with his PI Alkas and my PI Bogdan Pasanic. The results I just showed are current research. Thank you so much. If you have any questions.